La 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 la. Making sure my pen works. Looks like it does. Okay, welcome to adaptive, aka specific immunity 3, T cells. This is the continuation of chapter 16. So, remember the immune system can be defined broadly in two categories: the innate immune system or innate defenses, and the adaptive immune system or adaptive defenses. Adaptive defenses, it's also called the third line of defense. It's also called specific immunity. So you get 3 for 1 on that. But the major players in adaptive immunity are the B cells and humoral immunity, which we talked about in the previous video, and then the T cells, which do cellular immunity. And then we'll have video 4 and 5 for chapter 16, and then we'll put all the cells together and look at a synthesis of the complete immune system and how it works. So now we're just going to kind of talk a little bit about the T cells and what they do. Here is a cartoon of, can you guess what it is? It's a lymph node. You got it. Within a lymph node, there are these lymph nodules. And then within the lymph nodules, there are these things called germinal centers. And within germinal centers, that's where you find the B and T cells. So let's say there's bacteria or a virus somewhere in your body, and it is in your interstitial fluid. Well, if it's not in your blood vessels, it's in your tissue fluid. It can be taken up by lymph capillaries, which are right next to your regular capillaries. Lymph capillaries will take anything that it finds in that extracellular fluid and bring it into the lymph node. And as the lymph percolates through the cortex, then it will, in a afferent lymph vessel, so it's going toward the lymph node, and then an efferent lymph vessel will take it back out into the lymph system, and then eventually it will get back into the blood because it's fluid, so it'll get back into the blood. So this looks like it's going to be about B cells, but it's not. In most cases, B cells do not mount a humoral response directly. So if you remember the last video, B cells can recognize foreign invaders with their B cell receptors, and they can mount an antibody producing response, aka a humoral response. But most of the time, B cells need help to get fully activated to make their antibodies. So usually, B cells respond to antigens. Remember, antigens and epitopes could be found on foreign invaders. B cells respond only with the assistance of certain T cells. So that would be T-dependent immunity. And if you remember, T cells are formed initially in the red bone marrow with the B cells, but the T cells go on to mature in the thymus, and the B cells stay in the red bone marrow where they mature. Following maturation, either a B cell or a T cell, both B cells and T cells will circulate in the lymph and blood, and then they'll migrate to lymph nodes, the spleen, and Peyer's patches. So I like to use the cartoon, the lymph nodes, because there's lymph nodes all over your body, and this is where the B and T cells will set up shop to do their thing. T cells are very specific. Again, this is adaptive immunity or specific immunity. So T cells, like B cells, have a very specific receptor in their plasma membrane or their cell membrane, and that receptor can only recognize specific antigen/epitopes. So if you are a single T cell, you can have up to 500,000 identical T cell receptors sticking out of your cell membrane. And then the T cell receptors, they're proteins, so you got to start with DNA. T cell receptors are made similarly to how B cell receptors are made. We didn't really go into that with B cell receptors because it's a lot of genetic. All right, now we have to kind of take this, it's an odd turn, but we need to talk about these MHC proteins before we can talk further about T cells. So this is a cartoon. A is a cartoon of a class 1 major histocompatibility protein complex, or MHC class 1. And letter B, this is an MHC class 2 protein, and you can see whether you're an MHC class 1 or an MHC class 2, you're sticking out of the phospholipid bilayer of a cell. So if you are a class 1 MHC protein, you can find those sticking out of the cytoplasmic membrane or the cell membrane of any nucleated cell. And because I kind of like to do the military theme with the immune system, your own MHC proteins are kind of like your dog tags. They are unique to you. So all the cells of your body, all the nucleated cells, have your specific class 1 MHC protein in their cytoplasmic membrane. If you are an antigen-presenting cell, and antigen-presenting cells include macrophages, B cells, and dendritic cells, those are the major antigen-presenting cells, then you have a class 2 MHC protein. But this is a little tricky, so pay attention. If you are an antigen-presenting cell, you will have this class 2 MHC protein in your cell membrane. However, B cells, macrophages, dendritic cells, they are also nucleated cells. That means they have a nucleus. So, antigen-presenting cells have both class 2 MHC proteins, and they also have class 1 MHC proteins. Okay. So the function of MHC proteins is to present epitopes to T cells. So T cells and their T cell receptors will recognize epitopes / antigens that are presented to them in this antigen binding groove of the class 1 or the class 2 MHC protein. Okay, so we just talked about this, the classes of MHCs. Antigen presenting cells can be B cells or phagocytes. They present epitopes on MHC2 proteins to helper T cells. So that means this antigen presenting cell, if it gets infected or it phagocytoses a bacteria, that bacteria with its antigens and epitopes in its plasma membrane will get chopped up. So this guy got chopped up, and his or hers antigen epitope can get presented in this MHC2 complex, and the antigen or epitope, antigen/epitope will be presented to a helper T cell, and the helper T cell will recognize the antigen epitope with its helper T cell receptor. MHC1 over here are self-recognition proteins, so they can recognize self antigens, and then these class 1 can present abnormal epitopes to cytotoxic or TC cells. The T cell receptor specificity. So here's a T cell receptor. It doesn't look like a B cell receptor. Remember, the B cell receptor was a Y. This is a T cell receptor. And so it's in the phospholipid bilayer, or the cytoplasmic membrane of a T cell. It's got variable regions, and it's got constant regions, and it's got an antigen binding groove like a B cell, but it's not in the shape of a Y like a B cell receptor. So T cell receptor has only 2 polypeptides, 1, 2. But these terminal ends here of the variable regions will specifically bind to an antigen. And there's at least as many T cell receptors as there are B cell receptors in your body. And then this is just a cartoon of the B cell receptor, aka the antibody. Pretty much, B cell receptor and antibody is pretty much the same thing. All right, so let's take a look at this cartoon. Here's any cell. It probably should say any nucleated cell. Here's any nucleated cell's plasma membrane. Here's MHC1 from any cell. And what happened is this any cell was infected by some kind of pathogen. Let's say it's like a bacteria. And it's molecular machinery of any cell. Chop this guy up, and a piece, I can't really draw it like that, but a piece that looked like this was stuck into this MHC1 protein, and then the MHC1 protein and the epitope, a specific epitope from the bacterial cell, were presented outside of this cell. Let's say the cell got infected, and now the T lymphocyte. So this T lymphocyte. This is a cartoon of a cytotoxic T cell. So here it's T cell receptor of the cytotoxic T cell recognize the epitope in the MHC1 complex along with this helper recognition molecule called CD8. So now you have an infected cell down here, any cell, it's an infected cell. Here's what it's infected with. Chopped up the bacteria, presented the epitope from the bacteria here. The cytotoxic T cell recognized it with its specific T cell receptor that could only recognize this epitope here. And then it doesn't show it in the cartoon, but now that the cytotoxic T cell has recognized the infected cell, now the cytotoxic T cell can destroy the infected cell using perforins and granzyme to blow a hole in your infected cell and kill it. So the T cell, let's look at this stats here. It says the T cell receptor, that's this purple thing, does not recognize the epitope directly. So what that means is if this cytotoxic T cell here, this guy were just floating around in the blood, and this epitope came along, the T cell receptor wouldn't recognize it. The T cell receptor can only recognize this epitope when it's presented to the T cell receptor in an MHC1 complex. In this cartoon, the co-receptor, so here's the T cell receptor, and there's a co-receptor called CD8. CD8 is needed for a cytotoxic T cell to recognize and bind to MHC1. It's not shown in this cartoon, but CD4, so if this T lymphocyte were a T helper cell, it would have, its co-receptor would be CD4, and this NE cell would be presenting the epitope in an MHC2 complex. Let's see if I can erase all this mess I made down here so you guys can see that. Cool. So T cells, so we talked about cytotoxic T and helper T cells. act primarily against cells that have intracellular pathogens. So remember, if you're a T cell, you're looking for pathogens that are inside of cells. But cancer cells also have cancer molecules in their cell membrane that aren't normal molecules. So T cells can also act against cancer cells. All right, so let's look at this in a little more detail. Here is an infected cell. So this is any cell of your body. Check it out. Here came coronavirus, for example. Coronavirus came in, and then it doesn't show in this cartoon, but there's a whole bunch of molecular biology that goes on that we'll look at later that will chop up this virus into little pieces. And then they'll take one of those pieces of the virus. It will stick that piece of virus in an MHC class 1 protein and take the whole thing to the surface here. And now a cytotoxic T cell with its T cell receptor can along with its CD8 co-receptor, can actually recognize that viral epitope. So let's say this viral epitope originally started over here on the virus. So if this virus is just hanging around in the interstitial fluid or anywhere outside a cell, and this cytotoxic T cell saw it, it would not bind. That cytotoxic T cell would not bind to that epitope on the virus. If you are a cytotoxic T cell and you want to do your job, which is to kill infected cells, you need to see the viral epitope chopped up, stuck in an MHC class 1 protein, presented to the T cell receptor on you as a T cell. Then you can kill it. Then you can kill the infected cell. So yeah, there's no saving the infected cell. It's just going to get killed. Okay, cytotoxic T lymphocytes, or TC cells. Every cytotoxic T cell has unique T cell receptors. Yeah, we talked about that. As well as this glycoprotein called a CD8 co-receptor. So now you can call cytotoxic T cells, you can call them CD8 positive cells. So 2 names for one. Now once that immunological synapse is formed between the T cell receptor and the viral epitope in MHC class 1, then the cytotoxic T can kill the infected cell. It can also kill cancer cells like this too. So cytotoxic attack on the infected cell is performed by these CD8 positive cytotoxic T cells usually after co-stimulation with T helper cells or CD4 cells. And it doesn't show it in this cartoon. It just shows like this cytotoxic T can just kill an infected cell on its own. But most of the time, you need another interaction using the helper T cell to fully activate the cytotoxic T so that it can kill the infected cell. Okay, now let's look at this cartoon. So a macrophage is an antigen presenting cell. And at this end, it says the bacterial cell being engulfed. So this macrophage in this cartoon is not infected by the bacteria. This is really important to note. In this cartoon, the macrophage is not infected by the bacteria. The macrophage is actively phagocytizing or eating the bacteria. And you can see in this cartoon, phagocytosis, there's the phagosome and the lysosome fuse. You get a phagolysosome. And look here, you chop this bacteria up. And you chop it up and you get this piece, this red triangle. And it doesn't show it in this cartoon, but there is an MHC2 complex that's presenting this epitope to a helper T cell in this case. This is not a cytotoxic T cell. This is a helper T cell. And then the helper T cell will release molecules called cytokines that will fully activate this macrophage. So helper T lymphocytes are also called TH cells or CD4 positive cells. And it doesn't show it in this cartoon either. We'll have another cartoon with helper T cells that shows the CD4 positive part. So that CD4 is a glycoprotein receptor on their surfaces. So you'd have CD4. Oh boy, it's like I said, it's so hard to. CD4 would be like a co-molecule here on the helper T cell. So helper T cells co-stimulate other white blood cells like macrophages, but they don't do any attack on antigens or host cells. So remember, the cytotoxic Ts will actually attack and kill an infected cell, but these helper T cells just co-stimulate other white blood cells. They don't destroy infected cells. So helper T cells' function is to assist in regulating the activity of B cells, macrophages, cytotoxic T cells, by providing necessary signals and growth factors to fully activate those B cells, macrophages, and cytotoxic T cells. There's type 1 helper T cells. They assist in activating cytotoxic T cells and macrophages. And then type 2 helper T cells assist in fully activating B cells. And when I say this word activating here, the helper T cells activate T cells, macrophages, and B cells. That means they provide these signals and growth factors to the cytotoxic Ts and macrophages to fully activate them. Okay. So here's our helper T cell. Again, it says helper T cells secrete various protein messengers. Those are cytokines. So you've got the cytokines being secreted from the helper T cells. And cytokines, basically, we've talked about this in the past. If you remember, they're soluble. So that means they are soluble in the liquid portion of the blood. They are intercellular signaling chemicals. So if this cytokine is released from the helper T cell, then this macrophage will have to have a cytokine receptor in its plasma membrane to bind the cytokine. And these cytokines regulate the entire immune system, both adaptive and innate. Okay, so we've talked about cytotoxic Ts, helper Ts. Now we're going to talk about these guys, regulatory Ts, or as I like to call them, Tregs. That sounds funny to me. So here's a B cell with its B cell receptor. Here's a T cell. Here's a cytotoxic T cell, and they've all got their receptors here. Cytotoxic T cell looks kind of angry. Helper T cell looks kind of cherubic. And anyway, they're doing their thing. B cell, helper T cell, cytotoxic T cell. And you don't want them to do their thing without stopping. So you don't want the B cell to just crank out 1 billion antibodies and never stop. You don't want the cytotoxic T cell just to go around, lysing every cell it sees. You want to be able to stop that immune process. That's what Tregs do. They have a big stop sign. So they're also called suppressor cells. They repress the adaptive response and they prevent autoimmune diseases. So if these reactions, like the cytotoxic T cells, went around killing a lot of cells, it could result in an autoimmune disease. But Treg says stop, and then you stop. Okay, so we talked about this with the B cells, so hopefully you have kind of an idea, this concept of clonal deletion. So let's take a look at the cartoon. It says T cell tolerance, clonal deletion. Now, remember that the T cells are born in the red bone marrow, and then they go to the thymus to mature and become what's called immunocompetent. That means that they can go out and do the thing they're supposed to do. So this is what happens in the thymus with T cells. So check this out. Here's some immature T cells in the thymus. So look at them. They're all these blue circles. Those are the T cells. But then check this out. These green things, those are their T cell receptors. And look at it. So each one of these would have the exact, each one of these T cells would have the exact receptor all over its plasma membrane. So here's another receptor. Here's a receptor on a T cell. This is a funky little receptor on a T cell. So anyway, all these immature T cells, they've got their receptors. They're ready to go. They're ready to train to become mature T cells and go out in the world and do their thing. Now, in the thymus, they are subjected to these guys, ubiquitous self-proteins. So ubiquitous just means these self-proteins are everywhere. So these self-proteins could be found on any of your self cells. And think about it. You don't want your T cells to recognize your own antigens. So it says ubiquitous self-proteins, but they could also be called self-antigens / epitopes. So check it out. Here's the first T cell. Does this self-protein bind to a T cell receptor? No. That's good. I am a T cell. I used my receptor and I didn't recognize the self-protein. So now I am called self-tolerant. And there's a whole bunch more molecular biology, but this T cell can go out into the world and not have to worry, if T cells can worry, not have to worry that it will react with a self-protein. Same with this T cell. Oh, whoa, check out this T cell. It took a self-protein, and that self-protein bound to this specific T cell receptor. Sorry, T cell, you got to die because we don't want you out there recognizing self-proteins. Same with this guy. Recognition, death. No recognition, life. So, before you're even born, B cells and T cells have randomly generated all these different shapes of receptors on their cell surfaces. So every population of maturing lymphocytes in the thymus, if their receptors wrongly match up with a self-protein, they gotta die. And they die an apoptotic death, to be specific. It's important that we do this, that we do clonal deletion. This is the process, what it's called. It's important that we do this so that our immune response is not directed against our normal body cells. If that happens, that's an autoimmune disease. So the body will eliminate, they're called self-reactive B and T lymphocytes via clonal deletion. And clonal deletion, I told you, uses this process of apoptosis, so cell suicide. They basically kill themselves. Thanks, T cells. Here's a cartoon. This is how it's a little more complicated for T cell maturation. So here's a stem cell for T cell. It was born in the red bone marrow, goes out to the thymus. These T cells get their T cell receptors, and you see, and they all have different shapes at the tips. These T cell receptors will specifically bind an antigen or epitope, or it won't. So in this cartoon, it says here's the thymus cells. Here's an MHC1 complex. So the first question you have to ask, does the cell, the thymus cell, or I'm sorry, does the T cell, the immature T cell, does it recognize this MHC complex? Because remember, the T cell, with its T cell receptor, it has to be able to recognize the MHC complex. If it can't recognize the if the T cell receptor can't recognize the MHC complex, it can't form that synapse. So if that doesn't happen, the T cell can't even recognize MHC1, then it undergoes apoptosis. If you can recognize MHC1, like this T cell can right here. So here's the T cell receptor here. Here's MHC here with an epitope. Then that's good. This guy gets to survive. Next step, does this T cell recognize an autoantigen or a self-antigen? If the answer is yes, most of them will die an apoptotic death. If the T cell with its T cell receptors don't recognize an auto or self-antigen, then they get to be mature. Well, they get to go on to become mature T cells. And then even if you recognize an autoantigen, some of those T cells can become regulatory T cells. Okay, and I think that's the end. No, it's not. Oops. Clonal deletion of T cells, 3 things can happen. And we've kind of gone through this and talked about this. The cells, so these T cells that do not recognize MHC proteins, they're no good. They undergo apoptosis. Then if you are a T cell and you recognize an autoantigen, that's not good. Most of you die, but a couple of you get to go and train to be Tregs. And then the remaining T cells recognize, if you are a T cell and you recognize MHC in conjunction with a foreign epitope and not an autoantigen, so a foreign epitope antigen and not a self-epitope antigen, then you can be a immunocompetent T cell. You leave the thymus and off you go to circulate in the blood and lymph and hopefully find a nice lymph node to live in.